So it's always a good idea to write down the definitions of words when you see them defined on your screen. Okay, that's a good practice for effective note taking. So I'll read it one more time. Biotechnology is defined as an area of biology involving the use of living systems and organisms to develop or make products. There's an emphasis placed on genetic engineering specifically, so changing an organism's genome. So I'll give you 10 more seconds to make sure you have that definition written down. All right. So we want to think about biotechnology as trying to produce something, to produce a new outcome. And so here are some of the products that are produced as a result of our advances in biotechnology. Plant and animal breeding is one of the earlier, more ancient types of biotechnology. Our ancestors learned even thousands of years ago that breeding animals in specific ways gives them specific qualities. If I'm looking for a species, or if I'm looking for a breed of dog that has quality of strong scent, has the quality of high stamina, then I can breed specific types of dogs to produce that result. Okay, so that's an early example of biotechnology. What we see over here is, does anybody know what this is? This is actually a liger. So this is a lion that has been crossbred with a tiger. This is the offspring of those two. So there's a lion, potentially a lion father, and a tiger mother. So this is very uncommon. You wouldn't see this in nature, but we have learned how to do this in controlled environments. The interesting thing about this is that this liger is not able to reproduce. So we can't consider a liger its own species. In order to be considered a species, you have to be able to reproduce on your own. Ligers cannot do that, so they can't be considered their own species. Biotechnology has also given us vaccines. And of course, we're hearing a lot about vaccines these days as we are in search of a vaccine that would protect us from the COVID-19, the novel coronavirus. Cloning is also possible, but there are ethical issues and ethical questions that exist about cloning. Is it the right thing to do? Should we be doing it? We might have the ability to do it, but that doesn't necessarily mean that we should. Antibiotics, GMOs, which are genetically modified organisms, pest-resistant crops. So these are crops that insects don't really like to eat that much because they might be poisonous, or they might just taste bad, or they, make their digestive system out of whack. So these pest-resistant crops, eventually the insects will, learn their lesson, and they will stop eating the crops because they don't want to get sick or die. And so these crops are much less susceptible to being eaten before they can be harvested by humans and sold for a profit. Stem cell therapy, gene therapy, designer babies, that's an interesting ethical phenomenon because we now again have the ability to go in and edit a baby's genome and give that baby specific qualities that we might want to see. There are many others as well. We're going to talk about some of these qualities and some of these ethical questions on Thursday. Even lab-grown meat obviously has some ethical questions. We can now produce meat that, you might be able to tell a difference. It doesn't taste exactly like beef, but it's not nearly as environmentally harmful as beef would be. So those ethical questions have to be asked before we really start devoting a lot of resources to producing these things. Here's another definition for you. DNA fingerprinting is one of these types of biotechnologies that has become so important in the last 30 or 40 years or so. And DNA fingerprinting is the process of determining an individual's or a species' DNA. So this specific biotech allows us to establish a link between biological evidence and a suspect in a criminal investigation, or a person if we're trying to determine the paternity of a child, or even it allows us to make links between different species. So DNA fingerprinting is defined as the process of determining an individual's or a species' DNA. Does anybody need me to go back? Sorry about that. I'll give you about 10 more seconds. Okay. So DNA fingerprints are used to compare DNA. So if I collect DNA from a crime scene, and then if I also collect DNA from a number of suspects who are thought to have potentially committed the crime, then I can use this DNA that was actually present at the crime scene to pinpoint the person who is most likely to have left that DNA. We can use this, or we can do this simply by comparing the location of these DNA fragments. All right, so this is a process that we're going to talk more about in the next few slides, but we're literally just trying to figure out how many fragments the people have in common with the DNA that we collected. So in this case, we can see that suspect 2 has all of those exact same fragments. So suspect 2 is more than likely the person who left that DNA at the crime scene. So the way that we're able to do this, and again, it's all about, make sure you do write this down. The more fragments in common, the more related. So we're comparing these fragments. We're trying to count how many two individuals or two sources of DNA have in common. And the more that they have in common, the more related those two sources of DNA must be. The way we do this is through a process called gel electrophoresis. Gel electrophoresis. This is a process in which the DNA fragments are separated by size. Okay. So I'm not going to spend time on that because you all know it already. But we can apply this gel electrophoresis process to a number of different, scientific inquiries. So we can use it to identify suspects in a potential crime. We can use it to identify the paternity of a child, so figuring out who that child's father is. And we can also use it to catalog endangered species. So please do write down these three primary applications of gel electrophoresis. So let's try to compare some of the DNA that we see here. On the left side, we've got DNA that was collected at a crime scene. And then we've got a number of suspects who may have potentially left that DNA at the crime scene. All we need to do, excuse me, is compare the fragments and figure out which of these suspects has the most fragments in common with the DNA that was collected at the crime scene. So can anyone tell me which suspect that might be? Which of these four suspects has the most fragments in common with crime DNA? Thank you, Alyssa. We can see that suspect 2 has all four of those fragments that was collected at the crime scene, that were collected at the crime scene.
Yeah, it's 2.
So 2 must be the suspect, or 2 more than likely left that DNA, more likely than the other 3. Additionally, we've got another example on the right side. We collected more DNA. This is once it's been separated. So which of these suspects more than likely left this DNA? Thank you, Nick. Yes, 2. So this one's also pretty easy to see. You can see that the DNA left at the crime scene and the DNA that was collected from suspect number 2 is exactly the same. So it's more likely than not that this suspect left that DNA. Now, when it comes to using this for paternity purposes, it's a bit trickier. The reason for that is that, of course, children get half of their DNA from their mother and the other half from their father. So it won't necessarily be the case that the child gets all of these fragments from their father. Some are going to come from their mother as well. But we can figure out who the potential father is simply by tracing a line from these fragments all the way across and just seeing who has the most DNA in common. So let's take a look at this. So I draw a line through the first one. I can see that that same line is shared, that same fragment is shared by dad 2 and dad 3. I draw a line through another. This line is shared by dad 1 and dad 3.
It's 3.
And then I draw a line through the next one. Dad 3 also has this one. Excuse me. So dad 3 shares 3 fragments with the child. So dad 3 is more than likely the father of this child. Okay, so this is how it works. Here we've got 4 species, and we want to know which species is most related to species number 1. So this is a lot easier when we're in class and we can use rulers to actually draw lines, but this works as well. So we see that species 3 has 5 fragments in common with species 1. So they must be related. They're closely related.
Who's fucking racist that drives this truck? Huh? Who's racist that drives this truck? Oh. Who's fucking racist?
No way. Mr. Real, where you went to?
Oh, I'm back. Where'd the teacher go?
I don't even know. That's what I'm looking for.
Oh, this nigga just disappeared.
Yeah.
Oh shit! Nevaeh texted me. Oh, there he go.
Oh,
Where
yeah.
you go, Mr. Rudd?
Sorry about that. My laptop died.
I was
Yeah.
like, what happened? Are we done with class? I was like,
Okay, so I want you all to take some time to write down these steps. Please, I'm going to give you about 60 seconds to do that.
right
I'm
now.
going to mute everybody as well. Who's still unmuted? Oh, okay. Thank you for that, Brian, with your profanity. Someone else.
Yo, what we were supposed to be doing?
Okay. At this point, you should have had ample time to write down these steps. Now I'm going to explain them. So if you didn't write down, if you have not yet written them down, you should still have time to do so.
Wait, right?
Yo,
Oh gosh. Sorry
I
about
don't
that.
got no steps to write down. I'm over here rock.
Okay. Well, I'll take my time explaining these steps so that you all have more time to write them down. I apologize. I didn't realize I wasn't sharing my screen. But when we're trying to create a DNA fingerprint, obviously we need some source of DNA, whether this is saliva or blood or hair or bone marrow. We just need to be able to get DNA from something. At this point, we know that DNA is found in the nuclei of cells. It's found in the nucleus. So as long as we can find some remnant of a cell, whether that cell be a red blood cell or whether that cell be something that's found in the saliva or an osteocyte, which is a bone cell, we can extract the DNA from it. And that's the first step to creating this fingerprint. From that point, we need to be able to cut the DNA into fragments. So when we first extract the DNA and it is 3 billion base pairs long, it's not very useful to us. We need to be able to cut it up so that we can analyze it further. And by cutting it up, we can also start to determine whether or not one source of DNA is similar to another source of DNA. We can do this by using what are called restriction enzymes. We can think of these restriction enzymes as scissors, but they are scissors that only cut in specific places. So these restriction enzymes will recognize a portion of the DNA, and they will always cut at that portion of the DNA. They will always cut whenever they see or wherever they see that specific pattern in the DNA. Once the restriction enzymes have spliced the DNA, they've cut the DNA into fragments, we can separate the fragments through the process of gel electrophoresis, which we introduced on the previous slide. So the heavier or larger a fragment is, the less able it is able to move through the gel, the less able it is, the less ability it has to move through the gel. But smaller fragments are able to maneuver through this gel, and so they end up getting closer to the bottom. And I'm gonna show you an image that will help explain this more. We call it electrophoresis because it uses an electrical current. That electrical current is how the DNA is able to move. And then lastly, once I've gotten my fragments separated so that I've got the heaviest fragments at the top, I've got the lightest or smallest fragments at the bottom, then I can analyze it and I can compare my results. So I can say that this source of DNA has heavy fragments at the top, some fragments in the middle, some fragments in the bottom, and it looks almost exactly like this other source of DNA, which means more than likely these two sources are from the same individual, or they are from the same species, or this person is the father of this child, whatever we're ultimately trying to determine. So this is what gel electrophoresis looks like. And for those of you who are interested in studying science, maybe at a collegiate level, or if you're hoping to become a doctor, then you will have to take an introductory biology course. And you will also take the laboratory component of that course. And I can almost guarantee you that you will do this. So it's a very simple and very cheap procedure that is very common in introductory biology courses because it is pretty easy to do. But you literally, the first step is to extract DNA from a source. Then we cut up the DNA using the restriction enzymes. We then load the DNA into this electrophoresis tank. We just put the DNA into this tank here. And each tank is going to have a specific number of wells. So in this tank, for example, we've got 4 wells. So I can put a sample of DNA in this well, then I can put another sample in this well, and another in this well, and a 4th one in this well. Then I can turn on an electrical current, and that electrical current is divided up so that there's a negative side and there's a positive side. The positive side is going to pull the DNA towards it. If you've ever heard the phrase opposites attract, and I think we talked about this very early on in this course, and you'll talk a lot more about it in chemistry, but perhaps you've heard the phrase opposites attract. DNA is actually negatively charged, so it's attracted to a positively charged electrode down here. So the DNA is going to start moving towards this positively charged electrode. The heavier a fragment is, the less able, the smaller its ability to move through the gel. So your heavier fragments are going to be located at the top, near where the well starts. And the smaller fragments will be located near the bottom because they're able to move much further through the gel. Let's see what this looks like. So we're going to load in the DNA into the wells, and we're seeing how the DNA move further away from the well based on their size. We turn on the electrical charge and they just start moving through the gel. The longer, heavier fragments do not move very far at all versus the shorter, lighter fragments, which are able to move much further through the gel. So this process of gel electrophoresis is simple, it's cheap. It takes less than an hour from the time you extract your DNA all the way to the time where you're able to start doing some comparisons. And so this is why it's one of the first labs that you would do in an introductory biological laboratory class. So your restriction enzymes, they cut in specific places. They read the pattern of DNA, and they will always be able to cut in that specific place. So in this case, we've got a restriction enzyme that recognizes the pattern TAAT. So anywhere it sees a TAAT, it's going to cut right after that. So I've got AC, TAAT. Now it's going to cut right there. Then I've got GGTACAT, TAAT. So it's going to cut again. Then I've got CATGGTAAT. Then it's going to cut again. So it's recognizing this TAAT pattern, and it cuts there every time. And so what I end up with are fragments. So now this becomes a fragment of DNA. This is a fragment of DNA. This is a fragment of DNA. Here's another one, and here's another one. So we can see that there are different sizes. So this super long fragment here, in the process of gel electrophoresis, is not going to move through the gel very far. It's just too big. It can't move very far. But this really short fragment, which is only 2 bases, just C and A, it can move through the gel very easily. And so it's going to end up at the very bottom of the well. So the real life implications of this science and of these technologies has had profound impacts on our legal and criminal justice systems over the last 30 or 40 years. prior to the development of some of these technologies, our legal system relied almost totally on what's called circumstantial evidence. So evidence that is collected about a potential crime that is based on circumstance. Where was a person? Did anybody see that person in that time? Does that person have an alibi? Does that person have a known relationship to the victim of the crime? Does that person have a reason or a motive to have harmed that person? And so we relied on in some cases what some people might consider hearsay. But with the development of these technologies, we had a much more precise way to determine where people were and if they were likely the culprit of some type of crime. Now, this isn't always perfect. And as the technology has gotten better, we've also come to understand that in some cases it can actually do more harm than good. But in the case of this organization called the Innocence Project, it has done a lot of good in terms of freeing wrongfully convicted people from jail. So let's watch this brief 5-minute video. And then your asynchronous assignment for the day will be based on this organization.
You don't have that letter. Can I see that? This is it. You still have it. This is like, wow. This is, wow. Mr. Sheck, I'm confident that whenever the DNA test is done, it will show my innocence and this miscarriage of justice.
You're the best of the best when it comes to DNA evidence. This is pretty much known throughout the country, and I desperately need your help.
Greetings, I'm James Harden Jr. I'm writing you this letter in sincere hope that you could be able to help me and my brother, Jonathan Barr, regain our freedom.
My name is Jonathan Barr, and I was wrongfully convicted of a murder and a rape of my friend Katrisa Matthews, and I spent almost 20 years in prison for that crime.
I spent 15 years inside of prison.
I spent 11 years and 3 months in prison.
20 years, 9 months, 5 days in prison for a crime I didn't commit.
The system failed us, tremendously. It failed us.
What happened to this female is wrong, and the real rapist has confessed to attacking her alone. They allowed the victim to be shown mugshots. All the mugshots was in black and white with the exception of my ID. She basically focused on that, my ID picture, and when she saw me in the lineup, within 5 minutes after being shown mugshots, she identified me as her attacker.
It was a bite mark on the victim, and the odontologist said that my teeth was the one that made this bite mark.
There was DNA evidence that excluded us from being, the donors of the semen that was found inside of Katrisa. But I guess because they had these ironclad statements from co-defendants in our case, they still proceeded. We were prosecuted and found guilty.
I gave up in prison several times. I fought, I don't know how many appeals I filed.
I had just got denied parole for the 5th time, and one of the counselors that was working there, he pulled me. He wasn't my counselor. He just pulled me to the side. He said, "Look," he said, "Marvin, there's an organization out there that may be able to help you."
I had a lot of support from the Innocent Project. They were very, very helpful. That day I heard about the DNA testing and I was excluded. I was overwhelmed with joy.
It was a great relief to be able to walk out of there a free man after 14 years. I'm able to spend time with my grandkids. I can hold them in my arms. I can take them to church. Take them out to eat.
My mom was suffering from dementia and congestive heart failure. Despite of what her body was saying, her spirit was like, I can't die until my son gets out of prison. And she was able to see me completely exonerated on TV. And right after that, she died. She died at peace.
I wish my parents would have lived longer to see this, but I know in spirit they're here, and they will continue to be here for as long as I am here, and as long as my daughter is here. And if I am to have any more children, they will live through them. But I'm thankful. I'm thankful to be home.
I can remember my first day coming home from prison. I sat up and watched the sun come up that morning. And you see the sunlight every morning rise in prison, but to actually feel it, as a free man outside of the fences, it was a totally different experience. The whole time I'm watching it coming up, I'm saying, I'm free. I'm truly free. And without this organization, a lot of men and women who are free now would not have been free. And I am one of those men.
Okay, so please, again, make sure that you do dedicate time to completing the exit tickets every day. The exit tickets are very short exercises. They are not graded for accuracy. They're simply graded for completion. We use them as a means of reviewing the information that was introduced in that day's lesson. So please just dedicate those first 3 minutes of our asynchronous portion of class to completing that exit ticket. Then additionally, on Canvas today, your assignment is called the Innocence Project. So what you'll be asked to do is click on a link that will take you to the Innocence Project's website. On that website, you will choose a specific case, a case of a person who has been wrongfully convicted and since been freed by the Innocence Project. You'll read about that case, and then you'll summarize how DNA technology, how biotechnology helped free that person in 4 to 5 sentences. Okay, so that assignment is available on Unit 6, Day 1, where just under the warm-up that you completed today. We have 18 minutes left in class, and that should be ample time to complete both the exit ticket and the asynchronous assignment. So I will be here on mute if you have any questions.
Mr. Rudd,
Yes, sir.
in the exit ticket, he keeps saying something about a picture below, and there's no picture below.
Okay, thank you for bringing that to my attention. Let me check it out. Which question are you talking about?
So far, it was like that for question 1 and 2.
Okay. You may need to refresh because the images are there, and I can see it. I can see it in my student view of the quiz, so I know that they're there. Sometimes the images don't appear, but this time it does. It does look like it's there. So refresh, and
I just refreshed and it's still not popping up. It's like a little page, little thing, and then I'm trying to click on it and stuff, and ain't nothing popping up.
okay, give me a second. Okay, try now, because I just included the link to the image as well. So you should, if you refresh your page now, you should see a link. And that way, if the image doesn't appear, you can click on that link and it should take you to the page where we sourced the image.
All right. The Innocence Project is a grade, right?
Yes, sir.
All right.
Mr. Rudd,
Yes, sir.
the time ran out for the exit ticket. Am I able to get another attempt?
You should have another attempt. Yes.
It doesn't. I don't see where it say that.
Okay. It should be changed now, so you might just need to refresh your page or back out of the canvas course and try again. But it is now 1:20, so you all are good to go.
Okay.
Thank you for bringing that up, Anthony. And I will talk to you all tomorrow.
Okay. Let's see. Announcement. Assignment. Back into it.